Hi guys and Assalamualaikum. My name is Nuraniza Vidya Abdurrahman and today I'm going to explain to you about Kreb cycle and the steps of process. So let's get into it. Okay, first we get into the introduction of Kreb cycle. The Kreb cycle is also known as citric acid cycle (CAC) or the tricarboxylic acid (TCA) cycle. This cycle takes place in the mitochondrial matrix and it is primary step of aerobic processing within a cell. The purpose of the Kreb cycle is to collect high-energy electrons from these fuels by oxidizing them, which are transported by the activated carriers NADH and NADH2 to the electron transport chain. This is the Kreb cycle flowchart. Let's get into it step by step. Okay, first of all, we start with the glycolysis to Kreb cycle process, which is known as step 0. This step simply links glycolysis to the Kreb cycle. The pyruvate from the glycolysis will undergo oxidation, which oxidized by NAD+. It will form NADH+ as return and release carbon dioxide and combine with coenzyme and change into acetyl-CoA. Okay, let's get into the step 1. Step 1, the combination. So, the oxaloacetate, which has 4 carbon molecules, and acetyl-CoA, which has 2 carbon molecules, will be hydrolyzed by water and release coenzyme to turn into citrate. Step 2. Isomerization. Citrate undergoes isomerization and converted into isocitrate, which is an enantiomer of citrate. Okay, next we'll be getting into step 3, which is oxidation. Step 3. Deoxidation. Isocitrate undergoes oxidation and oxidized by NAD+ to convert into alpha-ketoglutarate, which is 5 carbon molecule. While undergo oxidation, NADH molecule is formed and carbon dioxide is released. Next, step 4. Deoxidation and combination. Alpha-ketoglutarate undergoes oxidation and oxidized by NAD+ to form 4 carbon molecule. After oxidation, coenzyme A binds to the succinyl and create into succinyl-CoA. The second molecule of NADH is produced and the second molecule of carbon dioxide is released. Afterwards, step 5, which is phosphorylation. Succinyl-CoA will undergo phosphorylation of GDP, which is known as guanosine diphosphate, and form into GTP, which is guanosine triphosphate. It is also to break down the bond of coenzyme A and change into succinate. Step 6. Oxidation. Succinate will undergo oxidation and oxidized by FAD+, and coenzyme Q will also involve in this step. The hydrogen acceptor in this step is FAD is because the free energy change due to oxidation is only high enough to able to reduce FAD but not enough to reduce NAD. So finally, the QH2 is released and succinate change into fumarate. Step 7. Hydration. So fumarate will be hydrolyzed by water and converted into malate. Finally, the last step, step 8, oxidation. malate will undergo oxidation for the last time and oxidized by NAD+, and then it will convert into oxaloacetate. By that time, the third molecule of NADH is produced. So now I'm going to tell you guys about the net output. So each cycle will produce 2 molecules of carbon dioxide, 3 molecules of NADH+, 3 hydrogen ions, 1 molecule of FAD2, and 1 molecule of GTP. Each molecule of glucose produces 2 molecules of pyruvate, which in turn produce 2 molecules of acetyl-CoA. Therefore, each molecule of glucose produces double of the outputs. Okay, that's all for my presentation. Thanks you guys for watching and hope you guys like it. And until we meet again, thank you and Assalamualaikum.